So I'd like to welcome everybody to the Science, Tools, and Technology presentation. We really appreciate the support from all of our sponsors. At this lunch presentation, we're going to ask we've invited our 5 gold sponsors to each give us a 10-minute presentation highlighting new initiatives and new results in their companies. So that's very exciting. And I think it really reflects how all through the AIR community development, we've had industrial sponsors and we've had research from industrial companies, very important help in the development of the AIR community. But every year we get more and more interest and support from industry. And I think that's obviously really important and where we have to go in getting the adoption of our standards and getting the word out there. So again, we're very excited to have this support. And our first presentation will be from ENPICOM by Nicola Bonzanni. And invite you to the stage. Thank you.
Thank you, Felix, and good afternoon, everyone. I'm Nicola Bonzanni from ENPICOM. And this afternoon, actually in the next 10 minutes, I would like to introduce you to our company, ENPICOM, and also show you why we decided to support this great community and perhaps give you a feeling of what we think are some great opportunities for the future. Now, in the past few days, it was really a pleasure to meet and see some familiar faces that I had the pleasure to meet in previous conferences. But for the people that don't know us, and it's the first time that we connect, I would like just to present who is ENPICOM. ENPICOM is a Dutch startup, has been founded in 2017, and it has headquarters in the lovely town of 's-Hertogenbosch, which is in the south of the Netherlands. And currently we are about 40 people strong team that work on developing innovative immunomics software solution for serving pharma, biotech, CRO, and the like. Now, more specifically, what keeps us really busy at ENPICOM is the development of our ImmunoGenomix platform, as we call it IGX. Now, IGX is a first end-to-end software as a service solution meant specifically to manage, analyze, visualize, and interpret immune repertoire sequencing data. And in most recent years, we have been working more closely to support antibody discovery companies. And some of the features that we have been developing the most are certainly a very advanced user interface and the ability to integrate multiple types of data, not only different types of repertoire sequencing technologies, but also all kinds of metadata that comes and can be associated with repertoire sequencing technologies. For instance, low throughput or high throughput cell screening, for instance, that is very important to be able to identify the best clones, especially when it comes to antibody discovery. Now, if we look under the hood, what actually does look like our platform? So our platform has a core around which a series of applications have been developed, and we keep developing and keep enriching this ecosystem. And the core is meant to provide data handling and all kinds of data management infrastructure, and around it, as I said, there are several applications, and each application performs kind of a different task. And we have applications, for instance, for annotation of clones, like IGX profile or MyXCR. And we have also application for quality control. We have application that provides some basic, let's say, repertoire sequencing analysis, like a VJ gene distribution and such. But specifically in the last couple of years, we have been working on 3 applications which are specifically focused for antibody discovery. Those are IGX cluster, IGX branch, and IGX annotate. And well, unsurprisingly, IGX cluster allows the ability to cluster B cells, IGX branch the ability to analyze phylogenetic trees, and IGX annotate allows you to include structural liabilities, prediction for structural liabilities, and developabilities to your analysis. Now, I don't have much time to show you what we can do or what our customer can do with our platform, but I just wanted to give you a quick bite of what that would look like with a short video that shows how certain part of the workflow, the discovery workflow can be conducted using our platform. And in this video specifically, we want to show how it's possible to visualize different clusters, select them, include some annotations, and then eventually look at the phylogenetic trees and such. So in this screen, you can see all these bars are different clusters that come from IGX cluster. Those can be annotated with a series of annotation that come either from a cell screening or from some other applications that are in the platform and generate metadata. Those can be added to the clusters. And when a cluster is chosen, then it's possible to visualize a phylogenetic tree, like in this case. And this is a phylogenetic tree that can be explored. It's very interactive. And very importantly, as you can see here, you can add annotation on top of the tree, like layering multiple level of data coming from different sources on the tree. In this specific case, what we are actually adding to the tree are liability annotations. So those are scoring that allow you to understand or allow our users to understand which clones actually have the most liabilities, which one doesn't have them, or which type of liabilities, as well as developabilities. And you can combine all these different annotation in a singular view that allow truly to explore the data in a very intuitive way and identifying the best clones for your follow-up analysis. Now, there are many other, of course, tools and possibilities within the platform, but this is not the right context to really show you all these different possibilities. What I would like to spend a few more minutes on is how we did generate these liabilities. So how do we come up with these predictions about liabilities and developabilities? And I think this is specifically interesting for this community because we did that in collaboration with the group in Oxford, the OPIC group in Oxford. And if you've been yesterday around here, you might have a chance to attend one of their tutorial in which they show their series of tools that can do all of that. And that's, let's say, the approach that we would like to take for these kind of things, because I think you guys are expert in your own field. And of course, you have very different, let's say, expertises, but you are the expert there. And what we bring on the table is our ability to create seamless workflows, the ability to put in the maintenance, the ability to put in, let's say, all the bioinformatics, software engineering combination. And this is the added value that we bring on the table in that kind of, let's call, partnership. And of course, we have also the commercial part on top of that. And this combination, I think, is very powerful and provides some opportunities for this community to valorize your work in the industry, also in commercial terms. And this is exactly what I think is a great opportunity for the future. So I'm sure, and I'm quite convinced that there are a number of groups here at this meeting that are developing extremely innovative methods or are generating extremely interesting data sets like germlines that could be used and could be applied immediately in the industry. And we would like to be the vehicle for that application in a sort of a collaboration, which is a win-win situation in which we're not the only one that benefit of that collaboration, but every of the two parties that collaborate into that have something to gain out of it. And they can put in what they're extremely good at. So basically bring in each one his own expertise and create win-win collaborations. So if you have anything like that, if you're thinking that you're working on the best nesting-class method for repertoire sequencing analysis for some part of the analysis or some innovative data sets, please reach out to us. We have a poster outside. We are also hiring 2 bioinformatics developers, so reach out. I will be happy to have a chat.